Hello, folks. So if you're out of time on the conference, all the industry guys, we basically know how to be in time with 10 minutes in the 30-slide lecture. So what's about PRECI? PRECI is a Ukraine-based translational science / DMPK company, which is busy with basically creating models for various kinds of diseases and normal pharmacological states that will assist in development and discovery of new drugs. So we've been assisting scientists throughout the world from already 4 years in various kinds of projects. And what should have been the brief, easy, light bulb project for several months turned for us into a 2-year journey. It's one of the directions that we're working currently, and it's a great honor for me that one of the heroes of this project are sitting here in this room. Maria is one of the project leaders on this beautiful adventure in extracellular matrix proteins. So why extracellular matrix proteins? Extracellular matrix proteins are not probably a bystander, but they're active witness of the diseases. So extracellular matrix proteins participate in regulation probably as no component in our homeostatic systems. So first of all, the regulation of integrin binding frequently dictates the signaling behavior in various kinds of ways. So there is no one way to describe how the ECM proteins work in the signaling. So there are a few examples on the collagen 6, for example, which should have been arguably the structural protein and the sole mechanical constituent, participates in so many ways in regulation pathways, starting from EGF to IGF to many GFs in our organism systems. But not only that, so unique modulus is of essential importance for the creation of an integrin synthesis. So we know that even small growth in the unique modulus of the oral system will increase the integrin synthesis by several orders. Therefore, integrins are regulators of the signaling, as it was said. And not only that, so structural proteins is one part of the picture, but ECM-affiliated proteins are a totally different part of the picture that frequently been avoided until maybe 10 years ago. So starting from matrix metalloproteases to transglutaminases to lysyl oxidases, those proteins have been known to digest or assemble the ECM matrix protein assemblies, decreasing the mechanical stiffness, therefore changing diffusion, changing the synthesis, changing the cytoskeleton form, inducing necrosis, inducing apoptosis, so different kinds of. So this dynamic complex system is abundant throughout the body, throughout any disease forms or any physiological states. So the question arises, how do we model it? So, okay, we can take recombinant collagen, we can take recombinant fibronectin, we can take recombinant laminin, and create hydrogel, create whatever mechanical structure we want to create from it. But we will never reach the complexity. We will never reach the particular complex system that we want to achieve. Therefore, the research in chaotic system, and we call it chaotic system because we never surely know the composition of it. We know only the abundance of the particular clusters of the protein present in this complex mixture. And therefore, this complex system, this chaotic system, has been recently popular. And our approach was to bring the material science gist into it and try to reconstitute, basically to create the hydrogel out of actual extracellular matrix proteins of the particular tissue. And that's what we started to do. So yeah, again, about the disease regulation. So our interest was driven mainly by oncology, since, in industry, oncology is growing. There are like maybe 7,000 new therapeutic modalities right now in development throughout the world. And our thought was, okay, we'll create the oncology models. But then just as we started, our customers started asking, okay, what about fibrosis? Yeah, apparently fibrosis is as well important in ECM remodeling, and ECM remodeling has been studied in fibrosis diseases. And actually, while in oncological diseases, ECM remodeling basically changes the structure of the extracellular matrix protein in reaction to certain stimuli. While ECM remodeling in oncological diseases is probably only sequence, basically a standard, a witness, a source of information for sure, but certainly not a trigger. Then in fibrosis, it's possibly a trigger. So what we know that fibrotic diseases are frequently regulated by myofibroblast differentiation. Myofibroblast differentiation is impacted only and surely by the extracellular matrix protein and cross-linking assembly yeah, basically all the kinds of. So what is an ideal extracellular matrix model? So ideal extracellular matrix model should be suitable well enough, first of all, to actually grow cells in it. As simple as it sounds, but it's true. So if you claim that you have a good extracellular matrix in vitro model, you should be able to grow cells. And second of all, you should be able to do proteomics. So proteomics been evolved with the Orbiter technology for several years already. So when we can capture not one, not two, but several hundreds of protein in the single scan and do Fourier transformation and figure out the actual composition of it. So if we target a new generation of extracellular matrix material, we should be able to combine both of them in a single system. So we have to have a system general enough to work with at least several kinds of tissues and tissue systems. Second of all, it should capture the primary structure of extracellular matrix as long as mostly correct folding. We'll never catch the correct folding. We need to be aware of that, but at least to try, at least to attempt to capture the correct folding of those proteins along with natural glycosylation, which is utterly important for extracellular matrix proteins because most of the mechanical properties, most of the colloidal proteins are determined not by the actual primary protein sequence, sometimes by the glycosylation patterns. All the extracellular matrix proteins have their own glycosylation pattern. And this actually defines what we will later see utterly important for actual modeling. So third of all, it should resemble the elasticity of the natural tissue, not too high, not too low, within the borders from 1 kilopascal to 30 kilopascal as in unit modulus representation. And possibly we'll need to manage to fabricate objects out of it. So basically, it should be semi-solid, ideally hydrogel material, which should work in vitro systems as it should. So earlier approaches were based on pepsin digestion of extracellular matrix tissue because those proteins are insoluble. Those proteins are starting from 100 kilodaltons and finishing probably with 1 megadalton molecular weight are practically, theoretically, nowhere soluble. So only way to solubilize them was to digest them. Okay, we'll capture some of it, we'll capture some part of the protein, but never capture all of it. So we decided to go a bit different approach. Okay, let's not try to solubilize it. Let's go into traditional polymer theory when we had two guys from DuPont called Florey and Fox, who told that the glass temperature transition, basically the crystallization pattern of the polymer, depends on the hydrophobic sites and the percolation sites and the zoophilic patterns of the proteins. Basically, simply saying that you should be able to create semi-liquid or semi-solid materials by increasing of swelling of those proteins, by increasing the amount of water inside of the protein structure, and later homogenizing it or doing whatever procedure you might want to do. And so when those proteins are saturated, probably we will have some procedure to remove water out of it and create some semi-solid material. So we went into deep forests of optimization. So actually the table is much contracted. So optimization of a correct swelling procedure required, I would say, more than 200 different reaction screening experiments. So we screened conditions. We looked for 4 major constituents. We looked for caloritropic agents as entropic stimuli to the protein water saturation. We looked for the temperature, which upon increase in the temperature, we know that the swelling increases, but what we didn't know actually, that upon decrease of the temperature, some of the folding occurs and the swelling also increases. So at +4, the swelling remains in suboptimal stages, whereas when you decrease the temperature to -10 or even -20, the solvation increases. Why? Because the removal of water because of hydrophobic effects is entropically controlled process. And entropically controlled processes are dependent on the temperature, and they the Gibbs energy of those reactions decreases when you decrease the temperature. So that's pretty fascinating thing. And surfactants, obviously, just not allowing protein to collapse. Sometimes needed, sometimes not, but we went without it because at negative temperatures, the system is viscous and stable enough, so the stability of formed colloids is enough not to append with any surfactants or anything from the external. And also reagents that remove some of the bonding, especially from the secondary structures, the disulfide bonding, which is pretty useless when you talk about still the proteins of several hundred kilodalton size. So decreasing the molecular weight even twice, it doesn't win you actually so much. So yeah, and then we form what we call ZOL. ZOL is semi-liquid stage which will spontaneously collapse into gel. It can happen very fast, it can happen very slow, but the process is going there. And we use ultrafiltration. So we use ultrafiltration to remove additional guanidinium molecules, whatever we added there, to create a pure ZOL gel system, which sometimes gelates, sometimes don't, but we know the perfect concentration at which it gelates, and we manage to form in this way the sol gel systems from various kinds of indication and nosologies. And then you can cryopreserve it, and so that's how we reach the product. What does the product give? So we started to investigate it. So atomic force microscopy is of particular use here. So what atomic force microscopy can give you, it combines the measurement of the size as well as measurement of elasticity of the particular particle. So what we see is the network formation, which is temperature controlled. So you can see the colloids remaining stable at I don't think you see the scale. It's 20 nanometer colloids, which begin to collapse when you increase the temperature to plus 25, and you already see some origins, some bursts of the networks at plus 25 degrees. But when you increase the temperature to plus 37 degrees, you can start seeing the perfect collagen stacking one with D bands, the characteristic bands for the collagen molecules, and you see the network. And then for the white gelation occurs. And what's more importantly, as we discussed at the beginning, the problem was not only to model the proteins, but model elasticity. So an elasticity can be modulated with this system using the centrifugal force controlling from 200 Pascal to 5 kilopascal tensile strain for particular gels using the simple procedures as centrifugation, just removing the extra water from the formed network. So in vivo, those gels didn't show any toxicity compared to any other injection systems like Matrigel or collagens, and resemble the same tumoral growth pattern with much faster onset of the vascularization in CD1 animals. So on hepatocyte models, so we see much higher albumin expression while using albumin secretion, while using the coating created from the liver extracellular matrix gels compared to other regular hydrogels like collagen 1 or any other.